So in this protocol extension, we created this new protocol to actually generate a strain-specific model from the reference model. It involves using a well-curated reference model as well as the genome sequences of other target strains within the species. It allows people to study the pan-metabolic capability and the strain-specific differences within the species. So it's a little bit different from the original model in the sense that it's actually semi-automatic. So in order to facilitate people to use this protocol, we actually included the Jupyter Notebook tutorial in the supplement for people to follow. Now I'm going to talk about the main stages of these protocol. This first stage involves using the well-curated reference model. So usually if a well-curated reference model is already being generated, you can find it from publications or through one of the websites from our lab, the big database. If the reference model has not been generated, you would then have to create this reference model from scratch, which following the original protocol, which may take up to 4 months. The second stage involves getting the strains of interest as well as actually creating the homology matrix. Usually you can get the genome sequences of the strains of interest from public databases or generated from in-house. One thing to note is that it's important to use the same annotation tools for the genome sequences to ensure consistency. And in order to generate the homology matrix, we use the bidirectional glass between the target strain and the base strain to actually find the homologous genes between the strains. And then the end product, as you can see in this figure, is the homology matrix that actually tells you the presence and absence of the reference genes within the model across different strains. The stage 3 would involve creation and functionally validating the draft strain-specific models. The creation of the draft models would involve using the output from stage 2, where you can identify which genes are missing from each strain, and you can delete the corresponding genes in reaction from the reference strain to create that strain-specific model. It's also important to update the gene reaction protein information within the strain-specific model. In order to validate the basic functions of these strain-specific model, you can then test the in silico growth as well as this biosynthesis of the biomass metabolite. If you do not see growth using these draft models, there are various ways you can troubleshoot and overcome these problems, including gap filling methods, alternating the biomass reactions, as well as supplementing the media. The last stage, which is stage 4, that's not included in the figure, is to manually curate these draft models. It's important to actually include those novel genes and metabolic reactions that are not present in the reference model for each strain and manually add in these genes and reactions. You can also follow the original protocol and look at the sections for refining the models to actually quality check and refine the models manually.